Okay, happy to see more people coming. Today is my last lecture for the semester, and we'll talk about the connection and how the advancements of representation learning and self-supervised learning translate to biological sequences. Of course, we need to focus on something, so we'll start on proteins because most of the research is happening on the protein domain so far. So let's start from a little motivation. I sent a couple of videos over to see what is a protein and stuff like that. We will not go very deep on analyzing what is a protein because what we really want to do is we take the sequences and we do some representation learning. That's what we do in this course. So a small overview, like why do we even care about that? So this is a really nice illustration from the David Baker, who has like the biggest lab on making research regarding generating novel proteins that are non-existing. So the the gray square indicates how little do we know, even from nature and all these generations of evolution, but there's still like an enormous search space to look for new proteins and new solutions. And on the other side is like some numbers from some video on YouTube that emphasizes the fact that many on the biopharma industry there is a huge investment and research on designing novel drugs and accelerating drug discovery. And this could lead to different drugs or vaccines coming up based on research on these fields, and this would be like the idea of adopting representation learning for such a new field would massively accelerate how fast we can narrow down all these space of possible solutions based on the models that we will see today. But before anything, we just need to take a small recap and focus on the attention because it will be the core part we will analyze today. So the transformer, you should always think the three dimensions of we have a three-dimensional tensor all the time. So we always have independent if it's a vision transformer or if it's applied in natural language or proteins. We always have three dimensions on the input. So bat size, L, we will call the sequence, so the number of tokens. We will see what is the number of tokens for us today. And the D model dimension. And the output follows exactly the same trend. So no matter how many tokens we have on the input, we get one output representation for each one of those tokens. We will ignore the fact that we have this classification and start of endings, and this will already included in this L. So the core attention operation, the self-attention, or in general attention, is defined as you can see on this by first applying this matrix multiplication between linear projections of the input. So we have Q, K, and V, which are the queries, keys, and values. So these are all, as you can see on the top, three linear projections of the input. And in case of cross attention, as on the image on the right, the query, the Q, is another representation, and keys and values always come from the same input. But Q can also be something completely different, as you can already see from the math. So this is the attention map. So for one head and one layer, this will be an L by L matrix. Okay, let's fix it. So this attention map will always be an L by L matrix, which means that without any post-processing, it already captures pairwise relationships. So we have a sequence of L tokens, and in the internal representation of the model already gives us like a pairwise relationship between how all these elements are connected to each other, which justifies its quadratic complexity inside this softmax operation, this matrix multiplication. And for those, just a small recall, this division with this with this factor is to scale down instabilities that arise from this large matrix multiplication. So I have a nice illustration here. So you can imagine that as here we have Q, you can imagine we have different embedding of tokens and K. So as this dimension of Q, so this D model dimension, as it grows larger and larger, this dot product becomes extremely large, which makes applying a softmax on top makes it a bit make this gradient saturate. We have analyzed again throughout the course. So this is a really nice illustration, and this is like the attention matrix and all the individual. So each row shows us how the different keys attend to this specific query. And the second thing you need to know is that you need to be conscious about is that all these, for all the input and output tokens with queries and keys, it's happening in parallel. So already this matrix multiplication computes all these pairwise relationship in one computation, one mathematical operation. So it's already seeing how all the possible queries map to all the different keys. And in the case of cross attention, queries and keys come from different inputs. So in language translation, the query is the decoding, the language we want to translate our text, and keys are the reference language, so translating from English to German and vice versa. And in the case of multi-head attention, for one layer, if we have 10 heads, we have 10 of those matrices. Is that clear? So for a transformer of 10 layers and 10 heads at each layer, how many attention maps can we extract for one input? If we have a transformer, we have 10 heads, and it's 10 layers, and each layer has 10 heads. So how many of those L by L attention maps we get from the whole model? Can anybody guess? 100, perfect. Because each layer gives us 10, and we have 10 layers, so 10 by 10, 100. Another thing that it's really, really important for today's lecture is the fact that these weights, so this attention map, which I have it here, it's that these weights are computed on the fly, or as they otherwise called in the literature, they are fast weights. Can anybody imagine why we call them like fast weights? How they are different from, okay, you can already guess from the illustration. Why I have colored the attention with different colors while the convolution and full connected layers are with black. Like what is the difference? Why we call them fast? Yes?
For example, in convolutions, the weights are shared between the kernel window. But here,
yes, you can already see this is like the kernel. But
yeah,
this just makes it local, but you can also make the attention local, but they still consider fast weights.
because they are not shared, they are generated like for every sequence or for every...
Exactly. So for each individual input sequence, these you get completely different weights. So they are changing on the fly. So even though the Q, K, and V, the W actually, WQ, WK, and WV are the weights, the linear projections. the attention weight changes for each input sequence, so you get a completely different image for each sequence. And this is not happening when you have fully connected layers or convolutions, where these weights just are slowly changing with gradient descent at each iteration. But once you train this model, you have the exact same weight. So this is a really nice advantage of having that I want to emphasize today. So for each input, you have a completely different pairwise relationships that you attend. And this happens L for M layers and H heads. It's like M times H. So because you have not just one attention map, you have for 10 layers, we said 10 heads per layer, we have 100 attention maps. So you can imagine how many of these pairwise relationships we have. Of course, in the first, let's say, layer, you have, if you have like, if the tokens are words, you have pairwise relationships between words, and in the second layer you have pairwise relationships of pairs of words, and so on, and you get to build more abstract representations. So let's take a look at what these attention maps look like in natural language, and specifically for language translation. One small detour is that this idea of fast weights and attending to all the elements of the sequence was already popular and quite effective from 2014 from this team about language translation. And here you can see in this square this example of these non-diagonal weights as indicated by the heat map. So the larger the weight on the attention map, the more wide the heat map becomes. So here you see this notion of order swapping that you need to translate from one language to the other. So this is from French to English, and you see the adjective and the noun. You may need to swap the order. So we're still staying in natural language, and we can already see, we will look at a lot of those heat maps. So don't confuse the L by L attention matrix with this visualization. So here, for each head on the x-axis and different layers, we will produce a heat map that corresponds to different things. So here we measure the confidence, so the maximum attention that each head at each layer, the attention that is assigned to the next token on the sequence or the previous one, it doesn't matter. It's like a way of reference. And then we take the mean over the whole test set. So that's why it's called mean, max, weight, attention weight. So everything, since you have the softmax, everything is from 0 to 1 all the time. And you can already identify some patterns. So there are, you can already identify that some heads, even in natural language translation, so this is still supervised training, we try to translate from one language to the other, and we see that some heads become extremely confident, and as we will see today, confidence is usually a very good thing in when you want to generate, like when you have a vocabulary of V elements, and you are very confident about your decision. This usually means that the model has materialized some sort of understanding for the domain you train on. So there is this work from some group in 2019 where they found a lot of, they tried to assign different roles to different heads learned for neural machine translation. And they identified some heads that just attend to their neighbor, so just one shifted value from the main diagonal, which are not really important. But the most important is the syntactic head, which tend to assign most of the weights into different relationship between language, so between noun and verb, or verb and or adjective and noun. So these are called syntactic heads, but they are empirically observed. And then there are some really rare and specialized heads, and you see that this is changing. As we said, these are fast weights. So these weights are produced for these individual input sequences. And these specialized or rare tokens give their attention to something that is really rare. So this word was really rare in terms of how frequent you can see this word, and you see that already some heads focus their attention there. And in their analysis, they found that all these the specialized heads, so this type of head is the one that you should look for, and they do the heavy lifting, while there are many heads that don't even have like a role, which are more or less uniformly spreading their attention, that can be just pruned if you want to create like a very fast model. So I will just talk a little bit about our sequences, which will be proteins during this lecture, but this applies to all different biological sequences. So the base unit are the 4 different nucleotides that can be found in our DNA or RNA. And then by considering all of the possible triplets, you get only 20 of those triplets are valid combinations that can be found in humans at least. So by taking triplets of nucleotide bases, we have the so-called amino acids, which for the purpose of this lecture would be the tokens or the elements, or otherwise called the residues of the sequence. You will see, based on different literature, different way to call them, but in practice this will be just, instead of words or path projection in vision, here we have amino acids, and this is like our basic unit that we will work with. And we will consider 3 different levels that you can work with proteins. So primary structure is when there are some ways in biology where you can do the so-called sequencing. So you see all these amino acids one by one with different biological techniques, which looks something like this. Here we have 2 chains of amino acids, and what we really want to see is how these are fold, and what is the outcome of the fold, which is the structures that they create. So they tend to create some patterns. For example, like these ones, you can see this is called beta seeds, and this is called alpha helices. So this is called the secondary structure, and which are kind of patterns that are localized and can be identified, and then you have the overall 3D structure, which is called tertiary structure. So during this talk, we will use the word interchangeably between amino acid, token, residues is a more biological, I just want to include it for completeness, or elements. And the vocabulary, which is natural language, would be how many words you have in your data set, like 10,000 or 20,000 words. It will be the 20 amino acids where you can, the ones that you can possibly have for proteins. If we had DNA or RNA data, we will have just 4 elements, which would be these A, U, C, G, these 4 elements. So you have a vocabulary of 4 elements. Here we have a vocabulary of 20 elements, and it's also called alphabet or lexicon in the literature. Then we will focus on 3 different things. So a very, very important thing about proteins is how different proteins or proteins with antibodies or different other molecules interact with each other. So there is a very specific part on the 3D structure on the protein that is responsible for connecting two proteins or a protein with another molecule, and this is called the binding site. So this is the part, the 3D part that we really care about, because if the binding site has a problem due to some mutation, then the overall function of the protein becomes useless. And in general, there is a very, very, very strong connection in nature between the structure and the function of all the molecules, like antibodies, proteins, it doesn't really matter, but let's stay on proteins. In general, nature highly correlates the structure, even on the human body, the structure of your lungs and intestines are really correlated with the function. So the binding site can make a protein bind to another micro or macromolecule, and this will cause a conformational change, which means that the protein will change its shape on the 3D space, which will result in a different function. So for example, you can think that people try to find like molecules, like pills or drugs or vaccines that bind in different ways on proteins and try to see if the reaction would be the desired one. In a more machine learning side, we will have an L by L matrix. So it's something completely different. Binding site is just a part that we really want to focus on. On the machine learning side, the target, as we will say, we have three types of targets. So in any case, it will be an L by L matrix. So the simplest case, let's say the most advanced case would be to have the exact distance in angstroms between all the different pairs. And you can imagine that since the distance is symmetric by definition, but we only need to have like the upper triangular or lower triangular to get this distance, because this matrix, in terms of physics, needs to be symmetric. So here, in the most ideal case, we would be able to predict the distance in angstroms between different pairs, and this would give us the exact structure on the protein in the 3D space, because if you know all the distances, there is a mapping with some advanced mathematics that you can go from this L by L matrix to the exact 3D structure. But since this is a bit hard to predict, because here you can see that the distances can have like many values, one thing we do is that the first simplification is to go from distances to histograms. So we beam from some range, we will see, for example, from 2 angstroms, or 2.5 I think, to 20 or 30, depends on the application. Let's take from 2 to 20, and then all the proteins that have like 1 angstrom difference are grouped together. So 2, 3, 4, and here we group together, we beam together all the proteins that have a distance between 2 and 3 angstroms, and this converts this very, very difficult problem where you have to predict the exact distance into a classification problem, and you have as many classes as beams. So this would kind of we would be able to formulate like a segmentation task where you have this pixel and you have to predict all the different classes. Is that clear? Okay. And then even more simplest case is to say that okay, I just want to create a binary matrix in the simplest case, where everything that is below a threshold, usually this threshold is between 7 and 8 angstroms, is considered to be a contact. So we consider that two amino acids are in contact if their distance is lower than 8 angstroms, and no contact if they are more than 8 angstroms apart on the 3D space. And then you can get a binary L by L matrix. So this will be the 3 possible targets. Let's say as a machine learning problem, we will always have the sequence, and we have these 3 possible targets, independent how much we want to find, like how much can we possibly predict, how much fidelity we can have on the predicted distances. And this substitution matrix, we'll see something you can get from bioinformatics based on different analytical methods. You can get like an L by L matrix for different proteins, and we will see the connection between the attention maps. So now we go back to the transformers. So we have the transformers, we have the input amino acids, and we want to see what's happening during pre-training. So now we ignore all the targets, so these contact maps and distance maps that are much more costly to acquire, and we stay just on the sequence alone. We consider that we only have the sequence available. What are the 2 possible approaches we have seen in the course for doing self-supervised pre-training? So we have seen the BERT approach, and a bit in smaller depth, we have seen this GPT, this generative pre-training approach. And why one over the other? So let's say we have 6 input amino acids, and we have a transformer, and in the end we get 6 output representations for those amino acids. So in the case of, let's say GPT, this generative pre-training, we provide all the sequence as the input, but the transformer has the so-called masked attention. Does anybody remember what is this masked attention doing actually? Yes?
So do you have to see the later tokens in the future?
Yes, so here if you're in position I, and you have the output token I, it can only see one side, as we say, like only the previous tokens. If we consider these directions, this output token will be the result of only the previous ones. And similarly, this token will only be the outcome of those ones. Is that clear? In the case of, so this is with masked attention. In the case of BERT training, we have as the so-called transformer encoder, and we don't have any masking inside the attention module, but instead we mask the input sequence. So usually 15% in natural language, and as we will see as well in proteins, we mask 50% of the sequence, and then we take the output representation that sees all the sides, so right and left side. That's what this B stands for in the BERT paper. This B stands for bidirectional, which means that this token can attend to all the tokens on the right and left side, while here these tokens can only attend to the tokens that exist on the right side. And so far, the biggest models in natural language adopt this paradigm, while in biology, so far, things may change, but this is the dominant type of pre-training. And here you predict the value with cross-entropy laws. So it's like a classification task in the end. And we have as many classes as the vocabulary, so 20 amino acids, 20, the vocabulary is 20. Any questions? Yes?
You do the same thing as in language where you don't always mask it, sometimes you switch it with a different model, sometimes you leave it the same. Or is it just the mask or not mask?
I think in here we just mask, but there are different BERT-like approaches. But I haven't seen this idea where you can provide like random stuff. I think you just randomly mask 15%. Any other questions regarding these two types and why we... Okay, I haven't explained why, but first, this is the dominant type in biological sequences and proteins specifically, and we don't want to lose by excluding some parts if we have like this biological sequence of amino acids, because we have no idea experimentally how what is the most important amino acid to predict this specific one. We don't know if looking backwards is a good approach so far, at least. So we want to include all the possible inputs, because the interaction between amino acids is highly non-linear and highly global. So even for the binding site, which I said that is like a very specific region on the 3D space, even if you mutate, so if you change somehow either with different perturbations, some parts that are completely irrelevant on the sequence, that could even correspond to, let's say, the other side, like the anti-symmetrical side of the 3D structure of the protein, it can even affect the binding site. So there are very, very highly global interactions. All right, so we have the so-called protein language models, we will use PLMs in short, where you have this BERT-like pre-training, and we will focus on that during the lecture. So we only have an encoder, normal attention, and then cross-entropy loss. So ideally, given that we have enough data, the model would be able to understand the interactions and the underlying principle based on evolution that led to this type of sequence to occur naturally. And another thing is that proteins do not need any input adjustment like vision. For example, in vision we said that we take these non-overlapping passes, but this is not the case during biological sequences, because proteins can be naturally represented as sequences. We don't have to devise about tricks, and specifically sets, because we said that transformers process the input sequence as sets, which is something that happens naturally in proteins, in contrast to even natural language, where there is a strong notion of order, and even on vision, where you have to or audio, where you need to define as some patches and then project them. So the fundamental question is, just by relying on the sequencing, so all these 20 different amino acids, what represent, and then doing this BERT-like pre-training, how much can you learn? What are the representations that one is able to learn and capture? So this is the first large-scale BERT-like pre-trained model, which is called ESM, primarily from Facebook AI, I think. They trained this model, and they find these properties similar to what we saw in the last lecture, how Dino creates these clusters throughout training, even though it's not trained with any type of label. Here you can see some properties of different amino acids and how they are clustered in a low-dimensional projection based on their weight and their charge. So you can already see some connection here. You can see negatively charged, positively charged, is hydrophobic, aromatic, and so on. So these clusters even emerge only from the sequence alone, and this is applied, I don't know if you remember, only in the output. So you only usually take one of the tokens, this classification token, which is like one of the tokens, and you make this projection. So how much data? Okay, we have only sequences. In bio deep learning, we don't only care about the proteins because we have 250 million proteins, which is, you probably think that it's a lot, but compared to natural language, there are very few. Not very few, but an order of magnitude smaller, at least. But we also care because proteins have a really varying length. So we really care about how many tokens we have. So we have 86 billion amino acids on those sequences. And it's really a big practical consideration and limitation how to process effectively sequences that have such a wide range of lengths. So a protein can have maybe a length of 50 amino acids, but even go to 2000. So you can imagine that zero padding a sequence from 20 or 30 elements to 2000 elements is not the optimal way to dealing with those data. We'll talk about the limitations in the end. But in any case, we see even from the first large-scale model that some really desirable properties emerge. So the model is able to capture, based on the output representation, some biological properties that are what we care about, and generalize across a wide range of downstream tasks. I will not cover it because it gets too application specific. And already we will see that it encodes information without ever being trained with any type of supervision on secondary structure or tertiary structure. It already captures some of those underlying information. Here is another visualization, again from the output of the transformer. So you can already see between an untrained and trained transformer how much the different, I think here we have like different genes and different colors correspond to similar groups that are called orthologous groups. And I've highlighted some clusters that you can already identify from a two-dimensional projection. And it's always desirable to see like tight clusters. So ideally, you would see like an ideal behavior if you have like 10 classes would have to be like, you have like tight clusters and very distant from each other. Have we done TSNE visualization on these largely clip models on the previous exercise? If you take a clip and you do this type of TSNE visualization, you will see almost a textbook exponential textbook behavior of an ideal TSNE behavior if you take, for example, cipher 10. So this would be the ideal behavior. All the Z groups would be grouped together and very far from each other with clear boundaries. But you can already see some properties related to that emerging. So to encode the secondary structure without going too much into details, they just add a linear layer on top based on the existing representation. So it's like a frozen, like no gradients are passed through the BERT model. And why, do you have any idea why they just want to see only with a linear layer how much of the secondary structure is encoded compared to what we do in vision? do you remember how we evaluate the pre-training division? We have some pre-training, and then we want to evaluate these representations. Yes?
We can do KNN or we can do linear probing.
Okay, yes. So this corresponds to some sort of linear probing. And do you remember why we favor k-nearest neighbors or linear probing over fine-tuning the whole network if we want to compare between different pre-trainings and architectures?
Because it's kind of fair if we are trying like different representations or different models, we try to measure the differences between the representations based on like a fair base.
Yes, so it's a more fair comparison because we keep the different representations fixed. So all the linear projection can only see, let's say, can only score as well as the existing representation. It cannot create a more abstract or more complex representation. And you can already see some linear behavior between the pre-training loss. C stands for cross entropy, I don't remember this E letter from. And this is the secondary structure prediction. I think these are 8 beams that they classified. What we said about distances, they just classified between 8 different beams. But you can already see that the better the model gets on the pre-training tasks, so this BERT, like pre-training with cross entropy, it automatically gets better. And I think the different colors represent different models. So then, I think 1 or 2 years ago, they scaled up this BERT pre-training. Again, no particular change on the transformer, no particular change on the data, just playing a bit with some hyperparameters and training strategies and training schedules. And they would be able to scale protein language models to 15 billion parameters. And they see that even more desired properties emerge. So here we have this L by L matrix, but this time it's not a symmetric matrix. So the lower right side is the ground truth. So these heat maps of blues indicate the contact. So what binds to what. So its position ij indicates that these 2 residues or amino acids are connected. And the upper left side shows the predictions of the model without any supervision, just by taking the average over the attention maps. So we just compute all the attention maps for this sequence and then take the average, and we can already see this information regarding the contact map to be already readily available for the model without any task-specific fine-tuning on this task. So this is really surprising, and that's why many people have switched and focused their research on biology and proteins and RNAs and all this data. But of course, as we have discussed, and you should always think in terms of representation learning, you should always think about the scale. So we have these huge data sets, but we also need a lot of compute and bigger models. And they also validated that, so keeping the data set fixed, they played with different number of parameters as indicated from the heat map. And this is the evaluation for contact prediction. This is another evaluation we don't care so much about. So you can see that the more the trainable parameters of the model, the better it gets, given that the data and the computer are fixed. So in large scales, you get the desired properties of learning underlying information regarding secondary and tertiary structure. And in this newest model of 15 billion parameters, They also saw, compared to another type of model which we will talk next week, that's called AlphaFold, that we don't need additional external information. We just take the sequencing as it comes from the reading the data. We don't need to find similar sequences on the database that you will see in the next lecture. And after they train the model, they use it for inference to existing protein sequences that they have been collected from nature, but we don't really know to which organisms they belong to. This is what they call natural metagenomic protein map. So they took like a huge database of 617 million sequences. It's just data that were sequenced, but we had no idea about their structure. And they perform inference. And as you can recall from natural language, we care not just about predicting the 3D structure with some modules on top of the language model that we will not cover, but we don't care about just the model predicting something, because this is sort of a generative task, but we care about its confidence on predicting something. So if you ever work with big protein language model or big natural language model, or in general generative models, this type of to perform this type of inference that generate all the tokens one by one. This is called autoregressive, where you take the output of your prediction and then. So in this scenario, you would only provide the first one, then you get the prediction, and then you feed the two ones. This is the so-called autoregressive inference or prediction. So when we want to evaluate language models or protein language models equivalently, we care about their confidence, and there is a metric for that if you want to look on the literature. It's called perplexity, which basically quantifies the confidence of the model and penalizes the model that provides for some tokens low scores. So a model that would output a trained model that would output a uniform distribution would have a very high perplexity, so it would be penalized, while a good model would have very high confidence, and ideally it will have like a perplexity of 1. And you see that